My name is Quentin Johnson. I'm a new postdoctoral fellow here at NIMBioS. I came from right across the street, basically out of Walters, made a short little trip over here. And today I'm going to talk to you about, as you can read the title here, utilizing computational methods to predict allosteric networks in protein complexes. So I started working on this talk a little while back, and I was getting into all the gritty details and doing all these figures, and then Sergey came by and said, you may want to make your talk a little more general. You know, I mean, not everybody is in your field, and it's kind of your first talk, so maybe more of an introduction might be more proper. So I took his advice, and that's what I'm going to give you today, something that's more of an introduction, talk about things that I do, kind of how the methods that I use work, and I'm going to give you some general background, try to hit all the different jargons and give explanations about the terms that you may not know. Okay, so let's step into it. Okay, so first thing I'm going to do is I'm going to give you a quick refresher on biochemistry. Of course, I'm not going to lecture to you, won't be a full class. Then I'm going to cover the tools that I use, which are chiefly molecular dynamics simulation, and also some data reduction and analysis techniques that I use, because MD simulation produces so much data. It's ridiculous. Then, of course, I'm going to touch on the mathematical and the computational aspect of what I do. Again, it'll just be a touch. I won't dwell on it too long. Then I'll get a little more personal, talk about biology that interests me. And then finally, I'm going to give you some real life, tangible examples of the things that I've done and what I plan to do. Okay, so let's start small, guys. All right, everybody knows this guy. Hold on, let me get this pointer going. All right, everybody knows this guy. This is a atom, right? It's got a nucleus with neutrons and protons, and it's got these electrons that orbit the nucleus. Okay, electrons are really important because atoms are able to share electrons. No, they're not selfish. They share electrons, and when they share electrons, they form a chemical bond. And then you start to see molecules, right? So everybody probably knows this one also. This is water. It's got 2 hydrogens and an oxygen. And as you add more and more atoms share electrons, and then more chemical bonds are formed, you get bigger molecules. This one here is a drug that everybody's probably on right now. It's caffeine, right? Anyway, so these molecules are very important, and people do study them, but I'm interested in bigger molecules. I like to study what they call macromolecules. And there are a couple of macromolecules that are quite essential for life. The first one, DNA. Everybody's seen the double helix before. DNA is basically the blueprint of you. Your body has this blueprint, and in your DNA, there's everything that you are. Your hair color, your eye color, the length of your legs, everything. Okay? So you've got this master blueprint. You're the foreman. You got this master blueprint. You want to disseminate that blueprint to your workers. But you don't want to give everybody the blueprint, right? Because if they tear it, now you have a mutation and something's going wrong in your body. You can't build the person correctly. So what you do is you copy that thing, right? You give a carbon copy, which is called RNA. Good thing about RNA, it's not an exact copy. It's a one-sided copy, but you don't need the other side because there is a matching of base pairs that goes on here. So you only need one copy. So it's a nice data reduction strategy by your body. Well, by everybody. So another one, a huge one that I'm interested in, just to say I really don't work too much with DNA and RNA. I have in the past, but what I really work on a lot is proteins. So I'm going to focus on proteins today, okay? So I think it's important for me to kind of hit this kind of central dogma of biology. You've got from DNA to protein. So as I said, DNA is the master blueprint for life. And you can transcribe or copy DNA into RNA. And this is just, again, a copy. And it's the blueprint for making the proteins. So now, one thing I should mention is DNA and RNA are codes, right? Like a computer code. You have a computer code in front of you. You may be able to read that code, but you may not see the process that code actually does. So you need to translate that code to get the actual thing that you want here. And that's what happens here in translation. RNA is translated to make proteins. Okay? So what are proteins? Everybody's thinking, oh, I had a nice steak yesterday. It's a great protein. Yeah, good protein in my diet. Don't get fooled, first of all. Animals are not the only things that have protein. Plants have proteins, bacteria have proteins, so on and so on. Anything that's alive, even viruses have proteins. Anything that's alive has proteins, okay? And proteins are really important, and there's a lot of biology going on, a lot of research going on proteins. Why? Because they are the most ubiquitous of the macromolecule. Well, I won't say most, but they are a ubiquitous macromolecule throughout biology, okay? So proteins have 4 levels of structure. We're going to start small. There we go. Which first is the primary structure. You can see here, they're like a beaded necklace, right? Each one of these little beads is an amino acid. You arrange these amino acids over and over again, and after a while, if you get enough of those amino acids, the proper amino acids together in sequence, you start to form what's called secondary structure. Now, what you're seeing, you can still see individual beads here and there, but what you're starting to see, what you concentrated on, is this alpha helix here. Is what's that called? What that is called. So there's alpha helices, beta sheets, loops, and other things, different secondary structures. This is kind of a coarse-grained way to look at the protein, right? So you can go even further to tertiary structure. Tertiary structure is a collection of secondary structures, right? So here you can see some of the helices here, so on and so on. And if you go even further, there's quaternary structure, which I won't go so much into that. Okay, so now let's get down to another important aspect of proteins. Proteins are able to bind small molecules called ligands. So if you think of the protein as this kind of Pac-Man looking thing, it's gobbling things up. This guy comes in, the ligand is this kind of triangle. The ligand binds to the Pac-Man, the protein, and now you have an active protein. See, before I had down here inactive, but with the combination of the ligand, it's able to do its process, okay? So we know from history that structure is really important for function, and structure is really important for the protein to be able to bind the ligand. So if you had the protein, the Pac-Man guy, kind of distorted, he's in a different conformation. You also hear me use that word conformation. Conformation is interchangeable with structure. Then now this ligand can't bind anymore, and you cannot get your active protein. So what a lot of what I do and other structural biologists do is investigate different structures of proteins in different conditions, because you are looking to either optimize or terminate that function, right? Okay. And also, what we're starting to see these days too is that dynamics are also important. So think about structure as like me standing here is my structure, and dynamics will be like how much fluctuation I have in structure right now. My dynamics are somewhat, and then smaller, and now larger, a lot larger. So dynamics are you can almost think of it how the protein moves, even though that's not the technical definition. Okay. So why do I care about proteins? Why are they so important to me? Because I like to call proteins the workhorses of life. They do everything. I mean, you think your bones and skin, all these things are doing everything. That's because they're protein. They have protein components, right? The proteins are the one that's doing the function for you, okay? So you have different types of proteins. You have structural proteins like collagen. Collagen is the chief component of your hair, your nails, your skin, your connective tissue, all the things that basically make up the structure of your body. Why collagen is such a good structural protein is because it likes to do what we call polymerize, which means add more and more of them together in a structured way, just like bricks, right? One brick is not so good, but when you put them all together, you form a wall, which is your skin, right? Also, there's storage proteins like myoglobin, which is important for oxygen transport. Also, you can think of hemoglobin is important for oxygen transport. So you're working out and you need oxygen. Myoglobin is taking that oxygen to your muscles. Hemoglobin is grabbing it from your lungs. Okay? Also, there's enzymes. Enzymes are really cool ones. Why? Because, okay, a lot of processes, a lot of chemical processes will happen if you leave two things that want to react together for long enough. But a lot of these biological processes need to happen fast. I can't just wait around for this reaction to happen. What enzymes do is they effectively speed up that reaction for you. So they lower the cost almost for you to do whatever reaction it is. And one particular enzyme I worked with is lipase. And what it does is convert fat to energy. It breaks down fat so you can use it as energy. You probably heard of like liposine or lipozyme, something like that. It's a drug out there in the market. And the reason it has that name is probably because it's derived either from a lipase or it's a drug that is specifically made to improve the performance of a lipase. Something of that nature, but that's why it's called lipo something. Okay. Okay, so proteins that I'm interested in, of course, I already mentioned the lipase. I did a study a while back with the lipase. Funny thing about a lipase is it's really efficient. When the ligand gets inside of that binding pocket, it does this reaction. But it likes to do the Pac-Man move, right? Open and close, open and close, open and close. So if the ligand's coming and it's closed, you never get that reaction. So we did a study where we categorized the open and closed states, and we were able to remark on the portion of the lipase that was important for giving you that open state from the closed state. And the whole thing was kind of a comparative study of that particular protein in different solvents, because the lipase is able to be influenced by its environment. Okay. Also, glycoproteins I've worked with, which are just proteins with sugars attached. The best example I can give for that are your blood cells. They have sugars attached to them. That's why with blood typing, sugars are really important for blood typing. Depending on what type of sugars you have attached to your blood cells, that determines your blood type. And that's the way that viruses, they trick your body. They have these sugars on the outside that kind of look like what your body's looking for, and then, they infect you, basically. And also nuclear receptors is a new class of proteins that I'm getting into. These are hormone-sensing proteins. They're very important for transcription. And they have, I want to investigate predicting this allosteric mechanism, as I said earlier, with computational tools. And we're going to get into a little later what allosteric is, because that's going to be important. Okay. So I normally describe myself as a computational biochemist. But you hear a lot of other people use a lot of other terms. Computational chemist is one that gets thrown out there. You hear a theoretical chemistry, theoretical biology, molecular mechanics. There's a lot of terms out there, but they all kind of have a common ground of they all involve some sort of computation to learn about or investigate some structure or dynamics of some molecule, whether it be a macromolecule or a small molecule or whatever else. Okay? Okay. So how do I do that? I use molecular dynamics simulation. That's the brunt of what I do, molecular dynamics simulation. Everything after that is kind of analysis of the data produced from the simulation. Okay? Okay, so this is a kind of large slide, so we're going to take it step by step. What I'm showing here is 3 different components: the initial structure, the force field, and the dynamics equations going into the computer, and the computer spitting out something which is called a trajectory. Okay, so let's take these things one by one. So the starting structure. Here's the starting structure of a protein. Actually, it's a protein complex. You got one protein there and one protein there, right? They're complex together. What you're seeing here is a secondary structural depiction of the protein. Again, you're seeing some of these helices here, and I think this is all the way helical. So you're seeing helices here and some loops and so on and so on. But this is not what the actual picture really is. This is for your eyes, your benefit. What it really looks like is something like this. It's an all-atom depiction of this protein. It's got every single atom shown here. You can see the greens are carbons, blues are nitrogens, and the reds are oxygen. Mostly it's green, right? Because we are carbon-based life forms. Okay? And so these sort of structures, you can display them as a picture, but what they really are is a file. Usually go by the name of a PDB. We have, go ahead and hit the next one. You have experimentalists that determine these structures, and then they deposit that structure online, and they call it as a PDB. You could find them at, PDB protein database. Okay, so what that file has is the X, Y, and Z, the 3-dimensional coordinates of every single atom in the protein. Every single atom. Okay? So that is very useful to me, because what I'm trying to do is to simulate the movement of that protein. And you can't do that unless you have some sort of starting point. Like, I'm not able to create that starting point with the computer. I need an experimentalist to find that starting point for me, and then I'm going to apply some of these things. It can come from crystallography. Mostly, I say probably 90% or more come from crystallography. Also, there's a NMR can do this. And what's X? I can't think of the name. There's another one that is not as high resolution, but can give you kind of a blob kind of picture of the complex. Yeah. So then now, okay, say we have this starting structure. Let's think of our starting structure as me, okay? And you want to know how that starting structure is going to evolve. Now, I need to know everything about me to understand what I'm going to do, right? So with atoms, what's really important is the force exerted on that atom by every other atom, right? And the way you determine that is with a force field, is what we call it, a force field. It's not something that shields you from incoming fire. It's a big mathematical equation. And what you're getting out of this mathematical equation is this guy up here, which is U, which is potential energy. That's the potential of some energy being exerted on that atom, basically, a force being exerted. Okay, so there's many different parts of this thing. Let's take it step by step. I've got this little animation here that is showing you some of the components. You've got a bond component, an angle component, and a torsional component. So as this guy right here, his bond is lengthening and shortening. That's where the bond component comes in. The angle here, the angle between that bond is changing. And torsion, you can think of as four things being connected, and I can't do my hand, but rotating about that middle bond. And you can see there's 1, 2, 3, I should go back here, 4, and this thing is rotating about that middle bond right there, okay? All right, so let's do one at a time again. Actually, we're going to do two at a time first because these two guys are really similar. They're both harmonic potential, right? So the math people already have noticed that this is some sort of harmonic term here going on. So what that means is I've got a little graph here to illustrate what I'm saying. You've got this L of 0. That's the length, the bond length, which gives you the minimum amount of energy, okay? So you've got this plot here where energy is on the y-axis, and the bond length here is on the x-axis. So you say at this R of 0, which corresponds to that L of 0, at that bond length, you have the lowest amount of energy in that bond. And as you increase that bond length, the energy goes up. Or as you decrease, the energy goes up, right? So it fluctuates around that energy minimum, basically. And the same thing holds for angles. It's just a theta here rather than an L, right? So for the torsional component, what you're looking at is a periodic cosine function. Rise and fall, rise and fall, rise and fall. Because you can imagine as this guy, I'm using the ABCD depiction here of 4 atoms. And I'm going to rotate about this BC bond, C and D. And I'm rotating towards. If you know chemistry, then this means that bond is coming towards you. So at first they were like this. Now I'm going to rotate, and now the D is coming towards you. So I've rotated about that bond, okay? And you can imagine, so this has got a similar energy as that, and this and that, similar energies, right? So that's why you got that kind of rise and fall, rise and fall. The last term is a non-bonded term, which actually is 2 terms in there. There's this guy, which is the van der Waals term, and this guy, which is an electrostatic term. Van der Waals is chiefly about repulsion, right? I like to explain this in a way of, okay, this graph here is showing you energy again on the Y and the interatomic distance, how close the 2 atoms are on the Y, on the X. I like to explain this like people. People and atoms are very similar. If I come too close over here to Sergey, a lot of energy being expended right now. He's like, get out of my space, right? Now, if I move over here, not so much energy expended, but we can still interact. I can still talk to him. Now, if I take a walk over to Hessler, there's no interaction at all, right? I'm too far away. And that's what's happening here. Too close, the atomic energy, interatomic distance gets too close, you start to repel each other. You get at an optimal distance here where the energy is at a minimum, and then as you increase that distance, the energy increases until it kind of tails off where there's no interaction at all, right? All right, so that's the force field. The force field is an explanation of all the forces on the atom, basically. So you know, so you have a starting structure. You know how the thing started. You know everything about that structure now, all the forces. Now the only thing missing is for you to explain how the thing is going to move. So you need the equations of motion. I'm not a mathematician. I'm not some advanced physicist, so don't expect me to derive all these things. But I am going to give you a quick rundown. I'm going to skip some things in between, but you will be able to follow the flow here. Okay, so we know this. force can be described in terms of an energy gradient, right? And this U right here is coming from that potential energy function we just talked about last time. So we have that, right? Now, thanks to our superstar scientist, Isaac Newton, we also know that force equals mass times acceleration, right? Now we have an idea about the motion of the thing, how fast it's going, the acceleration. But we don't want to know acceleration. We want to know the velocity, the change of the position over time, okay? So now, if you take acceleration, which is basically the change in velocity over the change in time, and you just move that m over there, now we've got it broken down to where we can see velocity, which is good because velocity is one of the things you're going to need as an input in your simulation. Now, we know velocity is what? Change in position, or position, change in position over change in time. That's what velocity is, right? So from the initial potential energy function, we can get all of this down to here, and finally we get what we're interested in, the change in position. This is my starting position. I have some interval of time, 1 second. This is my ending position. That was my simulation of me just now, right? But I'm going to apply that to thousands of atoms, basically. All right, so this is the overview again. We have 3 components that go in, put them in the computer, shake it up, press enter, and then you get a trajectory spit out. So what exactly is a trajectory? Trajectory is kind of a visualization of the motion of the molecule, and it's showing you steps in time on a femtosecond time scale. So you're talking about 1/4,000,000th of a second, okay? Now, each step of the simulation, each time the simulation iterates, you're going to get a snapshot. Snapshot sounds like it is kind of like what it sounds like. It's like a picture. But in that picture, you have a full set of atomistic coordinates. So you know the 3-dimensional coordinates of every single atom in the picture, okay? So I get a snapshot every time, every step of the simulation. So a trajectory is a collection of those snapshots, right? And it can be viewed like a video. I was going to embed something here, but I figured it would be easier and actually better if I just bring up what I want to talk about here, okay? Oh, that didn't work out the way I wanted it to. All right, so I guess we're going to skip that for now and come back to it. Oh, never mind. Here we go. All right. So I'm going to load, which I call the topology, which is telling me how everything's connected. And now I'm going to load, which is called the DCD, which is a trajectory, which is what I just talked about. And wish this thing would have stayed the way I had it. That's okay, though. Now, what you can see is this guy. This is my RXR-TR complex again, and I'm going to show him moving. Okay? And I'm actually going to give you a little bit better color representation. All right. So what you're seeing here is a RXR-TR complex, right? You've got one side right here is RXR and the other side is TR. I'm showing it in a secondary structural representation because if I show all the atoms, it just gets messy. Okay? Now, what some people are seeing here is the protein almost looks like it's breathing, right? Like it's going in and out, in and out, in and out. That's because the protein has a minimum energy conformation that it likes to be in, and it kind of just fluctuates around that well, basically. So think of for yourself. Your minimum energy conformation is you being at home, right? You fluctuate around your house. You go to the store, you go to school, but you always come back to home because that's where you're most comfortable. Unless you put in a whole bunch of energy to change that conformation to something else comfortable. You move, you get another job or something like that. Now you're in a whole nother minimum. Okay? That's the same thing with proteins. Yeah. Okay, so we're looking at right now picosecond snapshots. This is about 1 nanosecond, so it's 1,000 snapshots you're seeing over picosecond intervals. Right, so short, right? And we're talking about doing a nanosecond, which sounds like short to you, but for me it's kind of long. It takes a lot of work. The dimensionality, you mean like from side to side, how the scale? Yeah. You're talking about maybe in this system there's 500 residues, so 10 residues, 10 atoms per, or yeah, 100 atoms per residue. So you got like 50,000 atoms with each one has 3 coordinates. So then you're into the 150,000, and then you have a velocity attached to each one of them. And we're talking about like hundreds of thousands of numbers per snapshot. And I just said there's 1,000 snapshots you're watching right here. And my simulations run for probably 100 to 200 nanoseconds. So you're talking about like 200,000 times that 150,000. So it gets really, really, really big, really fast. And that's why I'm going to get into the data reduction tools, because it's hard to use your eye to kind of get some sort of meaningful, biological, it's hard to address some sort of biological phenomenon in a meaningful way if you're just using your eyes. You need a computer to do that, to show you the trends of the data and all these sort of things. Okay, I'm gonna go through this one real quick. So I don't use my computer personally. I use what they call supercomputers. So what is a supercomputer? So your computer is about, I'm gonna do comparison. Your computer is about 2 square feet. Supercomputer is about 4,000 square feet, right? Your computer has about 2 to 4 cores. This is a real supercomputer I'm talking about. It's called Titan. It's one of the fastest computers in the world. It's at Oak Ridge National Lab. Titan has about 300,000 cores. Your computer has about, if you're lucky, about 0.004 terabytes of memory. Titan has about 700 terabytes of memory. It can do about 0.08, again, if you're lucky, trillion calculations per second. Titan can do 20 quadrillion calculations per second. It gets to a point to where they just start calculating at what they call flops, which is floating point operations per second, I think. Yeah, per second. Okay, and then down here is a real life, this is not animation, this is a real picture. This is a real picture of Titan. This is the computer, which you can see. If you look here, this is a guy standing next to it. You can see computers stacked. So there's maybe, 20 computers stacked, and then I don't know how many rows you're looking at right here in columns. So it's kind of ridiculous. It's a whole room, bigger than this room, dedicated to this computer. There's nothing in there except this computer and fans to keep it cold. That's it. Okay. So all that amounts to Titan's about 2 million times faster than your computer. Okay, so why do we care about MD simulation? We got all these different experiments out there. You know, we got crystallography that get you really down to a small scale, and so on and so on. Because MD simulation gives you a spatial and a temporal resolution that you just can't get with other experimental techniques. I mean, you see the experimental techniques over here. These are different techniques: X-ray crystallography and EM and FRET and all these different things. And what the scales you're looking at here, or the axis here, is the length, the size basically. This is on nanometer length. And on the x-axis is time in seconds. So over here, X-ray is static. It can only see things in still pictures, but it can get down to maybe the angstrom level or nanometer level. But simulation, that's the best you can do. But simulation gets all the way down. It can see things that are happening at the angstrom level. This is 10 to the minus 1. Nanometers, which amounts to what, 10 billionth of a meter, and it gets all the way down to about the femtosecond time scale, which is a quadrillionth of a second. So simulation basically gives you the sharpest resolution on the spatial and the temporal side. Okay. Oh, okay. Yeah, I put this slide in because of me. Because about around this point, I start asking myself, okay, that's cool for you, Quentin. I'm glad you enjoy your research, but what does that matter to the rest of the world, right? So some real-life applications of this sort of science, computational science. Okay, first of all, you have the academic just kind of inquiries. You know, you want to know different things about molecules or physics or chemistry, and MD can solve those problems for you, give you a picture of these things in theory. Also, there's clinical research, which is pharmaceutical industries. It's huge, multi-billion-dollar industry. I was showing slides to high school kids last week about what I do, and I gave them a rather simple slide. I think the numbers on that was about 300 million you used in 12 years to make one drug, which is all fine if you make that drug and you get like $1 billion return. But what if you mess up? You just waste like 250 million, 10 years of your life, you know? Good thing about simulation, we can give you, so there's a lot of different drug candidates out there. simulation can help you pick the top candidates, so you're not sifting through 1,000 or 5,000 different molecules. You're working with maybe like 10 or 100 or 200, whatever. So it cuts that cost down exponentially, and it can cut that time down by a lot. Okay, also simulation is used as an experimental complement to NMR, crystallography, neutron scattering, things of that nature. A lot of times they use simulation to kind of fill the gaps in, because you can get a still picture with some of these experiments, but you can't get you can only get still pictures in certain intervals, so you can't see anything in between. And simulation can give you that in-between site. And commercial product design, I work at Oak Ridge about 2 days out of the week, and a lot of people there are working on biofuel research, so they want to figure out how to make different fuels using organic materials. And I work at the CMB, which everybody there not exactly does MD simulation, but they do something computational. Okay, so now we're going to get into one particular protein complex that I like to talk about, that's going to give you a little insight into what I like to do. So this is the nuclear receptors. We're talking about nuclear receptors right now, and nuclear receptors are essential proteins. They're transcription factors. They're able to turn transcription on and off via sensing of some hormone or some small molecule. These proteins are basically regulating gene expression. You can imagine if something goes wrong with this protein, you're going to start seeing it, right? First thing is you're going to develop hormonal resistance, right? The hormone's being produced in your body, but there's nothing there to sense it. So there's no reaction there. You don't get the reaction you're looking for. Then with that, you're going to start to see some pretty bad diseases. We got rickets, hyperglycemia, and the worst, of course, infertility. It's pretty bad. Nuclear receptors, they have a common core they have 3 common core domains. This is kind of the structure of the nuclear receptor in a cartoon. They have the DNA binding domain down here, the ligand binding domain. So this right here interacts with the DNA to help to do the transcriptional regulation. This part right here is able to receive the hormone, bind the hormone. That's the ligand binding domain. And then you have a linker in between the two that conveys that information that, okay, I bound my ligand, now you turn on transcription. That's the linkers. It's like a telephone cord. And so you frequently find these nuclear receptors. They're very social proteins. They like to dimerize, like to partner up. They don't like to be by themselves. So you find them as dimers, 2 of them associated together. And those dimers are able to display allosteric on each other. Okay, so now you're saying, okay, finally, tell me what allosteric is. Okay, I'm going to explain allosteric in the most basic example to try to keep it simple at first. Okay, allosteric basically means you have in context of proteins. You have 2 binding sites, okay? So you see these 2 proteins, RXR and TR. These little boxes here are their binding sites. See, the binding sites are not close together, but they're able to influence each other somehow. So there's this sort of, I got this dotted line to represent an invisible link between the 2 binding sites. So let's see what happens when RXR binds its ligand. Okay, RXR, that ligand out there, I'm sorry, I meant to mention, this is RXR's ligand and that's TR's ligand. So RXR's ligand comes out of solution and it binds to RXR, and you may not have seen it. Let's keep an eye on TR here. When that happens, you see the binding site of TR changed. It changed shape. Conformation changed. Those 2 things are not close together. How is that possible? RXR is signaling TR, okay, I've got my ligand guy. Now you need to change your binding site. Why? So now you can bind your ligand, because your ligand is not able to bind unless you get into that particular shape. And RXR is giving it that signal. It's time to change shapes. Okay? So that's allosteric. That's allosteric. 2 things are not connected, but they can influence each other. Okay, so why am I needed to detect this allosteric? It's been around for a long time. People know it exists. Why? Is because this signal that's being sent is happening on a angstrom type spatial level, and on a femtosecond to picosecond time scale, picosecond being a trillionth of a second, right? So you may be able to, with using experimental techniques, you may be able to get this picture, and that picture, but you can't get nothing in between because it happens too fast. And this is on a good day. Most of the time you can only get this picture and that picture, the unbound and fully bound, because that binding happened too quick for you to pick it up with crystallography. Okay, and NMR. Okay, yeah, so MD can be a way to connect those dots between the starting and ending structure. Okay, so particularly I'm going to talk about the RXR nuclear receptor complex. Again, that's just a depiction there. And we're going to focus on the RXR binding site, which is right around here somewhere. Okay, now I'm going to talk about the specific allostery that RXR TR is showing. This is called negative allostery by shifting the dynamics. Okay, so they found this out by experiments. I've collaborated with Dr. Fernandez over in BCMB. He actually worked on this directly. And he did an experiment where you have, as it's showing here, RXR and TR is a dimer. TR has its ligand bound. RXR does not. But they're titrating in some of RXR's ligand so that it will start to bind. And what they see is when RXR has its ligand bound, the electron density in TR's binding pocket starts to become discontinuous. What's electron density, Quentin? Think about it like crystallography is like you're taking a picture. Okay? Electron density is how sharp that picture is, basically. Take a picture of your friend and he's doing this right here. All this is resolved perfectly, but his hands are blurry. Why? Because he's moving too fast. The binding pocket is starting to move faster. The dynamics are increasing. The fluctuations are increasing. So I can't resolve that picture at the binding site. And that's happening as a result of RXR binding its ligand. But TR is not, it's not, those binding sites are not close together. That's why it's allosteric. Again, I'm going to just do it quickly here. Got the same setup, TR with its ligand bound, RXR does not have it bound, RXR binds its ligand, and now the dynamics of that binding pocket shift, hence the discontinuous line, and that makes TR release its ligand, can no longer hold on to that ligand because the dynamics, something in the dynamics caused it to let go. And we want to figure out how, why, all the things in between, right? So what we do is we take his initial PDB structure that he has, and we run a simulation on it. Okay, so there's two ways we could do this. One is we got one starting structure. Let's think of this box as all the conformational space. By conformational space, I mean every single conformation that this system can have is explained in this box. It all falls in that box. This is my starting structure. I start my simulation. I get another structure, which of course is close because my time step is very small. I can't I can't go but so far in a small step. And then another, and another, and more, and more, and more, and more, and more. Until I run the simulation long enough that I sample that conformational space well. Okay? It's fine if you, for my field, you need to get this sort of good sampling because it's not enough to say an event happened. You need to be sure that that event is happening. So you need multiple events. You need a bunch of events of this thing happening. And that can only happen if you get good sampling. You sample a lot of the conformational space. Okay? So this is the serial way to do it. I started with... Say it again?
What's the origin of stochasticity? What you showed looked like deterministic equations.
What do you mean the origin of stochasticity?
The randomness is coming.
Oh, where's the randomness coming from? How do I say this? Okay, so if you run a simulation, you would think if you start with the same seed, you will get the same answer. But that's not necessarily true. And I also thought that, and I said that in a group talk once, and somebody said, nope, that's not true. And I said, why not? And they gave me the answer, and I just don't remember. That's something I'll have to understand further. I don't remember exactly, because I have the same question. Where's the randomness there? Yeah, but I'm sorry I don't have a good answer for you. I'm sorry. So the other way to do this is instead of starting at one starting point and we're running one long simulation, I'm going to pick multiple starting points and run short simulations. Okay? So say my first system starts there. And I start the simulation. I run it. And I say, okay, enough with that simulation. Now let's push the system to a whole nother conformation that's far away from there, system 2, and run a short simulation. And then another, and then another. And then if you do that enough, then in theory, you will sample the conformational space in the same way as the serial simulation. But these simulations are not interdependent. So you don't need to wait for simulation 1 to be done for simulation 2 to start. You can run them all at one time. Okay? And I should mention here too, the different systems that we did here are mutations, point mutations of the RXR binding pocket. So that is intended to perturb the binding pocket in such a way that you will get sort of a loosening and this sort of beginning of binding, unbinding of the ligand and ligand to the ligand binding pocket of RXR. Okay? I know that sounds a little bit far, but just stick with me. Okay. So how do we find this allosteric network? Again, this is kind of an invisible signal sent, but there must be some sort of physical interactions happening that make up this network. And the way we thought about it was there are contacts being made between residues. So you've got two residues together, and if those two residues come close enough to contact each other, like right now Sergey and I can interact, then an interaction happens, some information is being exchanged. So we say, if contact is formed, yes, 1. If no, then 0. So it's a binary type thing, right? And we're going to do this for every single residue versus every other residue for every single snapshot, and then average over all the snapshots to get an idea of how often those residues come into contact, which residues are important to convey or send information left and right, or between the two proteins, is what I'm trying to say. Okay, so this is the first reduction of information that I'm going to do here. This is the first data reduction that I've done here. Why? Because you start with atomistic coordinates, 3 times the amount of atoms that you have. Now you're looking at residues, for one. There's only 500 residues versus like 20,000 atoms. Okay, so you drop your number down to 500. 500 times 500 because it's versus each other. So now you got 2,500, still less than 20,000. And you're only getting a 1 answer. You don't have a X, Y, Z. You only have yes or no. 1 answer. Is it 1 or 0? Okay, so that's that first. This is a way of coarse graining that X, Y, Z, that Cartesian information into contact information. Okay, so our two residues here, we say, I forgot to say my cutoff, you have a cutoff distance to say when they're able to contact each other, and I'm setting it at 4, and we say there's 3 angstroms between those two closest atoms of the residues. Okay, so they've made a contact. Yes, contact formed. So now we record that down here, 1 for yes, contact formed. That happens, of course, that table is much larger. It's over every residue. Okay. Still got a lot of data, right? There's still a lot of data because we're talking 2,500 times now 20 nanosecond simulation, which is 20,000 snapshots. So we're 20,000 times 2,500. Still a lot of data. But I would argue that contacts that are always formed or never formed are not important for conveying information, right? Think about yourself. You have a little shadow, a little kid walking around you all the time. You don't need to convey the information to him because he's there with you all the time. Or if you have a distant relative that you never call, you can't convey information. Y'all never interact, right? So it's only those transient interactions that are important because we're perturbing the system in such a way that we should see what we're looking for. So those interactions should form, break, form, break, form, break. Not just stay formed the whole time or not ever be formed. So that's how you convey that info. So what you're looking at here is what we call a rank contact curve, which is the contact probability that 2 residues come into contact on the Y, and just an index of all the residue contacts. The possible contacts. So 1 and 1 is 1, 1 and 2 is 2, and so on and so on. Okay. So up here, we've ranked them so that all of the ones that contact 100% of the time are at the left, and then it slopes down, and down here is a 0. So instead of taking all this data, we're going to chop off anything higher than 80% and anything lower than 20%. We're calling 80% or more always formed, and 20% or lower never formed. We're only going to concentrate on contacts in this transition zone, right? Okay. Yes. Okay. So I ran out of tricks to reduce my data and understand my data. So now I'm going to go to a more traditional data analysis tool, which is PCA. PCA, I'm not going to try to go through the whole thing. It's very complex, something you need to read up on if you don't know a bunch about it now already. But the gist of it is you have some data. The data is described by some axes. Say this is my data, and it's on these 2 axes. And I say to myself, man, I really wish that my axes were different so they could describe that data a lot easier on those axes. So if I could make me 2 new axes, I would put one there and one there. It's very easy to describe the separation of that data now. They're on a perfect axis, right? PCA is going to do this for you automatically in theory. Yeah. Okay, so now some data. What you're seeing here is a contact map of the PC1 contributions. PC1 is that top, highest, most important principal component, okay? And this is a contact map, so you're seeing every residue, 1 to 500 or 0 to 500 to 0 to 500 here. So you're not going to see in the diagonal because we just throw that information out, right? It's 100%. So I already said 80 to 100 is gone. 0 to 20 is gone. So you see a lot of white space here because there's a lot of stagnant contacts in the system, right? So we've gotten rid of those. That's the white space. Okay. So with the with this contact map, we're doing it in blue to red, basically, red to blue. So if the dot here between, let's say, 120, residue 120 and residue 70 or so, is forming a contact, then it's red. If it's breaking a contact, say between this residue and that residue, then it's blue. Okay. This is great, right? Because now I can start to see these clusters of dynamic contacts. Cluster here. There's a cluster here. There's a cluster here of dynamic contacts. These are places within the system that are important for conveying information. This is giving me an idea of what the mechanism is, right? How this information, the kind of line between the two binding sites. Okay? So again, this is this right here. This picture here is this picture projected onto a structure. So you see rods in between all the individual residues. Those rods are a direct depiction of the value, the PC1 value of these two. So say 300 and 320 have a red, then this is 300 and that's 320. There's a red rod connecting the two. Okay, and this is even better than this because now I can actually see on the structure where those little pockets of dynamic contacts are. So now I can start to kind of draw a line, which you can't really. I wish you could, but I wasn't able to actually show. Okay, here's the pathway, but evidence nonetheless. Good thing about this picture, this slide, is it's telling you, as it says, the contact map reveals increased dynamics at the TR binding pocket. Okay, TR binding pocket's right around here. You see a lot of strong red, strong blue, strong red, strong blue there. That means the dynamics are increasing. That means the contacts are formed and breaking very fast, because we're only showing the strongest, the most dynamic contacts, right? And that is supported by the experimental evidence, right? Earlier I said that when RXR binds its ligand, TR's binding pocket becomes more dynamic. That's what we're seeing here. Also, what we did is we took a sum of all the PC contributions of just the binding pocket with the ligand. So when the ligand forms contacts with the binding pocket, and what we saw is an anti-correlated relationship between the 9C and the TR, or the T3 ligands. So this is RXR's ligand showing a positive, which means the contacts are forming. The binding pocket, we're saying this binding pocket is a circle and they're all coming towards it. We're closing on it. So as this guy closes, TR is opening. It's letting go. That's the same thing that they were seeing here. It's the same thing we were saying here. That's a reminder of what they were talking about earlier, right? Okay, so that gives a little confidence in our method, but in the future, we need to get a much better method. I mean, this thing is so labor intensive. It's got codes and codes in different languages. And you have one person, me, that understands it all. It's no good. What I would like to do is develop a program that can automatically detect allosteric, and it basically be you as a user can upload your trajectory from your simulation, and it will spit you out something like that and that, those type of graphs automatically. So in the future, I would like to publish a methodology paper that details everything that is, make the program first, of course, and then publish a methodology paper that shows everything that I did and how it works. And now I'd like to have a kind of an online server where people can upload the trajectories and get back some info. That would be nice. It takes a lot of space, though, and memory to get the trajectories uploaded. So maybe it may be a downloadable program. Maybe that may be the way to go. And I would also like to apply the method to some other systems. In particular, RXR and PPAR, which has a positive allosteric. PPAR is very important in a variety of cancers. It's one of those proteins, if you can increase, if I can start the path to finding a way to increase this allosteric, that would be helpful for different cancers out there. Okay? And also there's ribose switches that are kind of the same sort of, not same, but they operate in a similar way. They detect something, and then they show a conformational switch at a site that is not connected to where the detection happened. So I may be able to apply this method to ribose switches since they're related. And also there's other sensing proteins out there that this method could be applied to. It's endless possibilities. Of that, I want to acknowledge Jeremy Smith, who is my advisor for NIMBIOS right now, Tonye Shin, who was my old advisor, Ricky, who was a postdoc in the lab, and Richard Lindsay, who was a technician now I work with. Elias Fernandez is a professor over in BCMB. He provided some of the initial structure for the ARC-STAR-TR complex, and also he got me interested in nuclear receptors. And of course, NIMBIOS for housing me, giving me somewhere to work and paying me. UT, because without UT, there isn't no one else, I guess. CMB, which is Center for Molecular Biophysics at Oak Ridge National Lab, which is the last guy. And that's it.